So remember, we're talking about this process called cellular respiration. And before you get all freaked out um, about the process, about maybe not understanding it, just remember that cellular respiration is a process by which it means it happens in cells. Every cell does it. Every eukaryotic cell does it. And it involves respiration, which is the use of oxygen. So, although we haven't talked about why oxygen is important yet, we did say that oxygen is an electron acceptor. And remember, this process of cellular respiration is basically using electrons to drive the production of ATP from the original molecule of glucose. And remember, it includes three major processes: glycolysis, the Krebs cycle or the TCA, and the electron transport chain. So far we've talked about glycolysis, we've talked about the pre-TCA reactions or pre-Krebs reactions, and we've talked about the Krebs cycle a little bit that happens in the mitochondrial matrix. But all of these processes are designed to make ATP in their own way. Some of them make ATP directly, and some of them make NADH, which is an electron carrier, which will then end up taking the electrons and using them to make ATP. But so remember, cellular respiration happens in cells. It's a way to make ATP. It occurs in the presence of oxygen, and it's it's basically a catabolic process. We're breaking down glucose in order for this to occur. Also remember that it's all about electrons. All right, it's all about taking electrons and using them for their energy. All right, so we start out with high energy electrons, and we're going to tired. Sorry. We're going to end up with low energy electrons. Happens in the mitochondria. Another important thing is looking at this molecule here. This is glucose. Let's see if we can. Can you describe if it's alpha or beta glucose? But this is just a glucose monomer, a sugar. Remember, where's the energy in that? Remember, the energy is in the hydrogens. And each hydrogen is holding an electron, and that's what we're actually going to be using to harness the energy for cellular respiration. So remember, cellular respiration is a process that happens in many steps. It's all about electrons. It's all about redox reactions. It's all about making ATP. It's all about harnessing electrons and using them for energy. All right, and it happens in cells. There's three parts of it. Happens both out of the mitochondria in the cytosol and in the mitochondria and on the inner mitochondrial membrane. So some places you should know are the cytosol of the cell, the mitochondrial matrix of the cell, the inner mitochondrial membrane of the cell, which is this interior black line here, all right, and the inner membrane space, which is actually in between here. This is in between these two black lines. This is all called the inner membrane space and the outer membrane of the mitochondria. So, remember, there's two major reasons why we do it in many steps. Number one is we can release more energy, or we can harness more of the energy by doing it in small steps, and we can also regulate the process by stopping if needed at a specific step. So those are two big reasons why many big steps is better than one fell swoop step. We're talking about redox reactions. What we're going to do is we're going to have electrons jumping from one molecule to another, like the hydrogens are going to get taken off. We're going to take the hydrogens and we're going to put them on something else, but the electrons are going to come with them. Remember, that's a redox reaction. When electrons, even in the form of hydrogen, jump from one place to another, that is redox, and that's how redox reactions are used to drive cellular respiration. Remember, the way we're going to use redox reactions is we're going to have things with a pretty low electronegativity down here, and as we move from step to step to step, we're going to be increasing the electronegativity of the atoms that are going to be receiving electrons. All right. Remember, one of the new molecules that we're talking about is called NADH, or NAD plus in its oxidized form. All right. We're going to gain, all right, 2 electrons and 1 hydrogen in the reduced form. And the oxidized form has basically 2 less electrons and 1 less hydrogen. So if you remember, we talked about NADH, this form of the electron carrier, only carries 1 hydrogen, but it carries 2 electrons, right? And where did those electrons come from? They came from hydrogens. So if this molecule can only carry 1 of the hydrogens and 2 of the electrons, it has to release 1 of the hydrogens in solution, all right, as a hydrogen ion. Remember, hydrogens have 1 proton and 1 electron. That's all they have, all right? So this molecule, NADH, is very important, all right? It is the reduced form. It carries electrons from one place to another. This is our electronegativity scale. It's just showing us the pull that each of these atoms has for electrons. All right, and remember, oxygen has a very, very high electronegativity, meaning it is going to have a very strong pull for electrons. All right, if we want electrons to jump from one place to another, if we had an electron on lithium and beryllium was close by, the electron would jump to beryllium. All right. If we had an electron on beryllium and lithium was close by, the electron would not jump to lithium because beryllium has a higher electronegativity. It's pulling the electrons towards it more. All right. And the same goes down the line. But oxygen, we're going to star this one as being very important because this oxygen is what we call the ultimate electron acceptor. All right. So a couple things we talked about. The ultimate electron acceptor in cellular respiration is oxygen. The ultimate chemical energy storage molecule is ATP, and the primary electron carrier is NADH. All right, there's some pretty important molecules there. All right, ATP, it contains a nitrogenous base, a sugar, and 3 phosphates. The energy that is stored in the ATP is stored between the second and third phosphate. All right, and that's where we're basically going to store all the energy that we take from glucose and the electrons and the hydrogens. We're going to store it in ATP, and if you remember, we're going to store it here so that we can actually use it for more things. All right, glucose can only be used for energy for a few things, but ATP can be used thousands of different ways. All right. Quick little review. We haven't quite talked about fermentation yet, but we will. All right. Mitochondria occurs both in animal and plant cells, but in all eukaryotic cells. The fuel is for cellular respiration is glucose. The role of oxygen is ultimately going to be to accept electrons. Remember, the whole process is all about the electrons and using their energy. We're using the fall of the electrons for our chemical energy in order to make ATP. We transport electrons using NADH. We store chemical energy using ATP. The reactions that allow electrons to move from place to place are oxidation and reduction reactions. So just a little overview. Again, remember there's 3 main stages of cellular respiration. There's glycolysis, Krebs cycle, and the electron transport chain. All right, we're going to kind of get into these now. Glycolysis is a process that occurs in the cytosol. It's a very primitive group of reactions, meaning reactions that basically have occurred since the first organisms could carry out these reactions. It starts with the 1 6-carbon molecule, and you end up with 2 3-carbon molecules. So glucose enters the cell either by diffusion or facilitated transport. In a bunch of steps, it gets broken down to 2 molecules called pyruvate. All right. Now, if oxygen is present, which is what cellular respiration is, oxygen present, the pyruvate is going to be transported into the mitochondrion as acetyl-CoA, and then it will enter the TCA cycle and then the electron transport chain. But a new thing here is that if oxygen is not present, all right, then fermentation is a process that basically uses the pyruvate in order to create some energy from it. But again, oxygen cannot be present in fermentation. All right. So we're going to be talking more about fermentation, but for now, we're going to talk about glucose to pyruvate, pyruvate to TCA, and TCA to the electron transport chain. All right. If we go to glycolysis, I don't want you to memorize any of the reactions. I don't want you to memorize any of the enzymes. I don't want you to memorize anything, but what I do want you to do is understand why each reaction is important to the process. All right. So we talked about hexokinase is an enzyme that catalyzes the phosphorylation of glucose here, puts the phosphate on there. And the reason why that's important is to keep glucose in the cell, number one, and also increase the energy state, the instability of glucose, to make it more reactive. All right. And this reaction, all right, happens spontaneously, and it uses an ATP to occur. This second reaction changes from glucose to fructose, same number of carbons, hydrogens, and oxygens and phosphates, except you can see there's a different structure. When there's a structural change, all right, an isomerase enzyme is always involved. And this structural change, this process occurs in order to make the glucose molecule into fructose, which is more symmetrical. Because if you see in the future, we're going to split it into two. So we want a molecule that's more symmetrical so that we don't have to change it around as much. As you see, the enzyme being used here is a kinase, so you know the ATP is involved in some way. All right. Again, an ATP will be used, and its phosphate will be put onto the fructose. This makes this molecule more unstable, more reactive, number one, and secondly, it makes it more symmetrical. All right. This next reaction here splits this molecule into two, all right, which leaves us with two similar but not exactly the same molecules. But in order for this to be the most efficient process, we take one extra step and we change this molecule into this molecule right here. All right. Now, everything that happens from glyceraldehyde 3-phosphate on, you need to remember, happens twice because there's two molecules of G3P, glyceraldehyde 3-phosphate. All right. This next reaction is really important. It produces our first electron carrier. All right. So we're going to get a hydrogen from solution. All right. And we're going to get a hydrogen from the molecule itself. All right. So we're going to create a situation where we make an NADH, which is going to go to the electron transport chain. And we add a phosphate onto the molecule, but realize that we didn't use an ATP in order to do this. So this is very beneficial. So up till now, up through step 6, we've used 2 ATP, and we've created 1 NADH per glyceraldehyde 3-phosphate, but there's 2 of them. So we've created 2 of these. All right. Now we put a phosphate on this molecule without using one, which is really helpful. And remember, a dehydrogenase enzyme is one in which NADH is involved. So if you see NADH involved in a reaction, you know it's dehydrogenase. If you see a dehydrogenase enzyme, you know NADH is involved. All right. So which brings us to our next step, step 7. All right. Step 7, you look at the enzyme. It's called a kinase. That means ATP is involved, except now we're taking a phosphate off of the molecule, putting it on ATP. So now we've produced 2 ATP. So we've netted 0 so far because we've used one here, we've used one here, but now we've gained 1 per molecule. So we've gained 2 here. So now we're even. All right. Remember, step 1 through 5 is called the energy investment phase of glycolysis, all right, because we use energy to get the molecules that we've made here, all right. This step 6 through 10 are all about taking energy that we can use later on, either NADH or ATP. It's called the energy payoff phase, all right. So we've created 2 ATP in step 7, all right. We mutase changes groups, so we're going to switch the 2 and the 3 functional groups here. As you see, they're switched. This next reaction takes out water. All right, water is a byproduct or a waste product of glycolysis. And our next step here, our last step, pyruvate kinase. Again, we take our last phosphate off of phosphoenolpyruvate, put it back on ATP. So now we've created how many more ATP? 1 per molecule, which means 2. Great. So we ultimately end up with 2 pyruvate molecules. All right, as you can see, there's not that many hydrogens left on them, almost completely oxidized. And pyruvate now is going to be traveling into the mitochondria for these reactions that we call the pre-TCA reactions. All right, so generally glycolysis occurs in the cytosol. It starts with glucose. It ends up with 2 pyruvate molecules. It produces 2 NADH. It produces 2 net ATP. All right. Great. If you have any questions, I'd love to answer them for you. That's just the first round of cellular respiration. And remember, if oxygen is present, pyruvate will then travel to the mitochondria for the pre-TCA reactions. If oxygen is not present, then actually cellular respiration cannot occur. So the pyruvate will go through a process called fermentation, which we'll talk about a little bit later.